Hello, everyone, and welcome to Dana Farber's Department of Data Science Frontiers in Biostatistics webinar. My name is Erica Fike, and I'm the Program Manager for the department. If you have a question for the speaker or the organizers, please use the Q&A tab, or you may raise your hand to ask your question live. Our faculty organizers will manage the Q&A session during and after the talk. With that, I turn it over to Svetlana Tykucheva.
Thank you very much. Thank you all for joining us. Today, we're very fortunate to have Dr. Elizabeth Garrett-Mayer as our speaker. Dr. Garrett-Mayer earned her PhD degree at the Department of Biostatistics at the Bloomberg School of Public Health at the Johns Hopkins University, and then joined the faculty of that department, as well as the Department of Oncology at the Sidney Kimmel Comprehensive Cancer Center at the Johns Hopkins School of Medicine. Later on, she joined the Medical University of South Carolina, where she was a Biostatistics and Bioinformatics Division Director in the Department of Public Health Sciences and Director of Biostatistics for Hollings Cancer Center. In 2017, she joined ASCO staff, where she until recently led Biostatistics and Research Data Governance Division as a director. And today, Dr. Garrett-Mayer is a vice president of the Center for Research and Analytics at ASCO. Throughout her fruitful career in oncology biostatistics, Dr. Garrett-Mayer co-authored nearly 300 peer-reviewed publications, co-edited 2 books, and wrote 9 book chapters. And today she will tell us about early phase design considerations for oncology drug development in the era of immunotherapy and targeted agents. Thank you.
Okay, yeah, thank you for that great introduction. It's great to be here with you today. It's nice to see some familiar names among the attendees, and thank you for inviting me. It's a great pleasure to be here. So if you can see my screen, you can see the title of my talk. I changed it just a little bit, and I'll explain why in just a moment. So I am, as was stated, Vice President of the Center for Research and Analytics, which is the CENTRA division at ASCO. So I've been at ASCO for about 4.5 years. Since I arrived at ASCO, I've become a bit less of a methodologist and a little bit more involved in meetings with stakeholders and other people interested in cancer care, optimizing treatments for patients, and helping care providers provide the best care for their patients. So my talk today will be a little bit more, I hope, thought-provoking and thinking about ways that we as statisticians and researchers can help move the field forward, and a little bit less about the actual statistical methods involved. However, I think that having statisticians and other methodologies at the table is really important to understand these issues and to speak to other stakeholders about their importance. All right, so I'm going to start out with the 2 points I want to make. So I'm unfolding my story, but at the same time, I'm letting you know where I'm going. So 2 points I want to make today are that the commonly used dose finding designs do not find optimal doses for targeted and immunologic cancer agents. And second, for many currently approved anti-cancer agents, dose optimization needs to be revisited. Right, to the overview of my talk, I'll give a historical perspective on dose finding in oncology. I'll keep this brief. I'm going to give a couple case studies of what were considered phase 1 studies of immune checkpoint inhibitors. I'll talk about revisiting dose finding objectives. I'll talk about dose optimization in the post-market setting. And then I'll talk about some other opportunities that I hadn't touched on at that point for trial designers and methodologists, for those of you interested in moving this field forward as well. Okay, so what are the dose finding methods in cancer drug development? I think there's a pretty diverse audience here. I know there are currently about 40 attendees, some of whom may be familiar with dose finding methods in cancer, some of whom may not. So the traditional phase 1 dose finding approach is toxicity-based dose finding in the cancer setting. So here there's developed in the era of chemotherapeutics and cytotoxic, the idea that the more drug a patient can tolerate, the better in terms of having an impact on the cancer. And there's an assumption made that as dose increases, toxicity increases, but also as dose increases, efficacy increases. And that has been really 2 main assumptions that have gone into statistical design assumptions about phase 1 dose finding designs. So the common ones we see, the most common, unfortunately, is the algorithmic, the 3+3 design, which has maintained its abundance due to it's an easy and people understand it. So here's just a little schematic to show you, if you're not familiar what the 3+3 design is. It's algorithmic without any real statistics involved. You enter 3 patients at a low dose level. Depending on how many patients have an adverse reaction, quantified a dose limiting toxicity, which would be defined in the protocol. You either escalate to the next dose level, you enter more patients at that dose level, or you stop. And then this algorithm continues until you find an optimal dose where either 3 or 6 patients are treated at each dose level. In 1990, the very important sort of seminal approach, the continual reassessment method, was introduced by John O'Quigley and others. And also later than that, the EWOC design was introduced, which represents escalation with overdose control. Other methods have been based on probability intervals for doses, the MTPI and the Boyne designs, and likelihood-based dose selection. So here's an example that's a little bit more rigorous than the 3+3 design that shows for acceptable and unacceptable dose levels, depending on how many patients are treated at an individual dose level, what the likelihood ratio would be in terms of evidence for or against that dose level being considered acceptable, where green means it's an acceptable dose level, red means it's unacceptable, and the grays and the pinks and the light greens representing weak evidence of acceptability of the dose. Here we see a schematic that shows what the CRM design looks like in practice. There's an assumption of an underlying dose toxicity model. Data is collected and a model-based design is used to estimate the toxicity level at each dose, and to also to assign patients to doses, the next patients that are enrolled in the trial. Right, so those are that's what's done in a traditional phase 1 setting. Of course, things are not always so simple as having a single agent. We have combination drug trials very commonly in cancer, where we're looking to identify MTD for both or at least one of 2 drugs. There are some model-based designs. Nolan Wages has spent a lot of time on the proportional odds CRM, and this shows the way that he approaches it, where you assume that that there are 2 different drugs, drug A and drug B, and that you understand that there's an ordering that goes up horizontal or diagonally in this direction, and in this other opposite diagonal direction, you have you don't understand if these doses, which one would be more toxic than the other in this direction. But in this direction, you can say that doses here, D5 and D2, would be more toxic than D1, for example. And this is more efficient than simply just escalating each drug in different directions. There have been a lot of work on delayed toxicity events. The time to event CRM was developed by Ken Chung and further developed by others. The rapid escalation design, the red design, developed by Ivanova. This shows an example of the idea behind some of these designs, where you expect that toxicities will be delayed. These are commonly used in radiation oncology trials, where sometimes there are late toxic effects, and you don't necessarily need to wait a specific time to collect patients' information, but you treat their toxicity as a time to event endpoint instead. And then a lot of these studies will also incorporate expansion cohorts. Historically, expansion cohorts in traditional phase 1 designs have been geared towards further confirmation and understanding of the safety profile of the MTD, the maximum tolerated dose that was identified in the escalation portion of the trial. Alexia Iasonis has done some work where you can use expansion cohorts to recalibrate the MTD, recognizing that oftentimes we have relatively few patients treated at the MTD in the dose escalation part of the study. Okay, so I want to move on to talking about defining toxicity outcomes. In most of the traditional phase 1 studies, there is a binary endpoint called the dose-limiting toxicity, or the DLT, that's defined in a protocol. This is something that is not consistent across protocols. It will depend on the expected toxicities associated with the agent under study. So this will be defined in consultation with the clinicians involved in the study, pharmacologists, and others to state what would be considered an unacceptable toxicity or a dose-limiting toxicity. These usually are grade 3, 4 toxicities graded through the common toxicity criteria. There has been a lot of work in recent years recognizing that a single binary representation of a patient's toxicity or adverse event profile on a phase 1 study does not really well encapsulate their experience. So work has been done on developing ordinal grades for toxicity, thinking that that would have a little bit more sensitivity and information in it than a simple binary endpoint. And then also toxicity burden scores. The idea here is that patients can have multiple different toxicities, and having just one DLT versus multiple of them potentially should have more weight and be represented as more serious than having a single event that would count as a DLT. So Nate O'Connell, Xing Li, and others have spent time working on these toxicity burden scores that of course have to be done in consultation with clinicians to understand the severity of different adverse events and how they would contribute to an overall score. One of the things that we have a challenge with sometimes in these studies is attribution of the adverse events, whether or not the treatment under study is the cause of some of these adverse events. Sometimes it could be due to another aspect of either the patient's cancer care or the cancer itself, and the relatedness, which is, I guess, all wrapped up in the same thing. And then another thing we have a real challenge with sometimes is in a lot of dose finding studies, we really only focus on the adverse events that occur during the first cycle. This doesn't really fully capture the experience of patients as they stay on drug for more than one cycle. So oftentimes we'll see delayed events, or we'll see potentially cumulative toxicities occurring, and they may not be incorporated in the dose finding component if the definition of a dose limiting toxicity really only reflects the first cycle of treatment. And just for reference, a cycle of treatment usually is usually 21 or 28 days of therapy, but it can vary outside of that. So there has been work done in this area looking at post hoc evaluation of toxicity in later cycles. So Xing Li and Javier Paoletti both have nice work looking at that, where there might be an MTD defined in the phase 1 study, but at the end of the study, you can actually incorporate information from across the whole study and recognize whether or not that is really the appropriate MTD, or should you be using a different dose going forward for future clinical trials. Okay, and then also we very often see in the early phase setting, phase 1/2 designs, historically 2 separate phases within one protocol, usually answered sequentially, where we have first the phase 1 question is asked, what's the correct dose? Followed by the phase 2 question, is there sufficient efficacy at the selected dose to warrant further study? The historical benefit of this, doing them in one protocol, a lot of it has been really to avoid having to have 2 different submissions and 2 different approval processes. So what that means is you would have to go through a scientific review for 2 different protocols versus one. You'd have to go through IRB review for multiple of them, and then there's just a lot of sort of red tape involved with getting 2 protocols up and running versus simply one. For several decades, authors have been working on a combination of efficacy toxicity designs, where either simultaneously or sequentially you're using model-based approaches to address both toxicity and efficacy. And this is really sort of the direction to move in the sense that, you know, the idea of finding an optimal dose without incorporating efficacy does seem a little bit naive. Unless you can make that assumption, that as toxicity increases, so does efficacy. It really doesn't really make sense to base dose finding purely on a toxicity outcome. So there's been lots of work done here. I won't get into all the details. There are some authors listed here, but like I said on the slide, this is really an incomplete list. Okay, so in short, we have a lot of designs. They can do all sorts of things regarding targeting an optimal dose based on one or more criteria. We do have a problem in the field that there's a low uptake of new designs, and we still have a focus on toxicity, even when we have good evidence from preclinical or other drugs in the same class to suggest that toxicity-based dose finding is not optimal. So why have we not had uptake, and why is there this focus still on toxicity? Well, a lot of these newer designs are more complicated to implement. The newer designs require more patients to get an answer, so they are seen as less efficient from a resource perspective, yet they are more efficient, certainly for getting the right answer. The 3+3 designs and other algorithmic designs still are well accepted, despite the fact that their properties have been shown to be very poor. And a lot of this has to really just do with the idea that, change is hard. Most of the dose finding community is used to seeing 3+3 designs, and there's not an awful lot of pushback about it. So these designs tend to be perpetuated, although there are better options out there. Okay, so I want to go through a couple case studies. These are two agents that were received breakthrough designation, and it was very clear that in this class of drugs, the approach for phase 1 dose finding was not appropriate. And some of you might know these stories. I find them really interesting, and a lot of times statisticians don't really know the background with some of these the drug development and some of these studies in pharma. So just a little bit of background about breakthrough designation. In July 2012, the US FDA, there was the US FDA Safety and Innovation Act that was signed, and this included a new designation for an experimental treatment called breakthrough therapy designation. And a breakthrough therapy is defined as a drug which is intended alone or in combination to treat a serious or life-threatening disease or condition, and for which preliminary clinical evidence indicates that the drug may demonstrate substantial improvement over existing therapies on one or more clinically significant endpoints. So it has to be in a serious or life-threatening disease, which of course cancer is one of them. And of course, in terms of certain areas of advanced cancer, there can be few treatment options that are successful for a number of patients. So if a drug receives this breakthrough designation, then that company whose drug it is, or whoever has the drug, the FDA will expedite the development and review of such drug. And what that means is that the trial that it is already ongoing will evolve in consultation with the FDA to move it through to receive approval. Okay, so the immunotherapy breakthroughs that I'm referring to, most of you have probably heard of these drugs, Nivolumab, which trade name is OPDIVO, and pembrolizumab, whose trade name is Keytruda. These are immune checkpoint inhibitors, and they block a protein called programmed cell death, or abbreviated PD-1. And these PD-1 blockers free the immune system around the cancer by helping T cells to attack the cancer. So both of these drugs were in phase 1 trials years ago. In version 1 of their protocols, the sample sizes were 32 and 76 respectively. Both of them gained breakthrough designation, and the companies worked with the FDA to expedite development. Okay, so what I'm showing you now is if you look at the pembrolizumab protocol version 1, this is what the study looked like. It had 30 patients with advanced solid tumors, and it had 5 dose levels to which patients were to be assigned. When all was said and done with this protocol, at the end, it ended up with 1,235 patients. because it received breakthrough designation. So in the early phase 1 part, there were some really astonishing responses seen in some patients. And then you can see, if you look at this slide here, that not only were there disease-specific cohorts, you can see melanoma is included here and non-small cell lung cancer. But if you look closely at the different little boxes on here, what you see is quite a range of different doses. So throughout this breakthrough therapy, working with the FDA, became very clear that the optimal dose, the dose finding, was still ongoing with, hundreds up to over 1,000 patients at many different dose levels, ranging from what you can see 10 milligrams per kilogram Q2 week 10 Q3, 2 at Q3, etc., for all these different cohorts, randomized and non-randomized. And the point here is that clearly the dose finding that is going on here, the optimal dose finding, is not based on the toxicity, whereas here in this early phase of the trial, this is just based on a simple 3+3 design, trying to find the optimal dose. So that was pembrolizumab. So for nivolumab, the first version of the protocol occurred in July of 2008. There were 3 dose levels, 1, 3, and 10 mg per kg, a 3+3 design with a planned N equal 12, and then some expansion cohorts. And when they got to protocol version 5, 4 years later, not quite 4 years later, you can see that the additional doses that they added were quite a bit lower than the ones that they had in this initial version 1 of the protocol. So they were added in Amendment 4, and they had up to 14 expansion cohorts added as well. And at the trial's end for nivolumab, there were 296 patients enrolled across 5 cancer subtypes. So this shows from the Amendment 4 to the protocol what they were adding. And at that point, this here shows these were already completed cohorts. And again, you can see the range of doses considered. And you can see over here, after these 1, 3, 10, and 10 mg per kg, protocol amendment 4 is looking here at 0.1, 0.3, and 1, and 3, of course, as well, and 10. So these examples really serve to show us that from the very beginning, the dose finding here wasn't done appropriately. Because these studies did receive breakthrough designation, the FDA worked with the companies to try to do a better job at dose finding. And these are now, very successful drugs that are given to many patients with cancer. Okay, so now I want to talk about revisiting the dose finding objectives. So like I noted earlier, we've historically believed a model that looks like this, and this has to do with the fact that cancer drug development really was very much focused on cytotoxic treatments. And here we have response shown in red. So as dose increases, the probability of a response should increase. And also as dose increases, the probability of DLT should increase, dose limiting toxicity. And the idea being that the more you can give a patient, the higher the response rate is going to be. But what we see now, and this is a bit of an exaggeration, is relatively flat dose response curves and relatively flat dose toxicity curves. Now, this one here shows very low toxicity. The toxicity rates may be higher in certain agents, but there's not necessarily a very strong dose toxicity relationships in terms of having, a steep slope or a slope that is non-zero. And the same thing with response for some of these agents. Speaking of which, for the Pembro and Nivo, the original dosing was in mg per kg, and they've been converted now to flat doses. So regardless of weight, patients receive a fixed dose, just like, with an Advil, you get, a fixed dose of Advil regardless of your size. Okay, so revisiting the objectives. In traditional cancer treatment, the dogma has always been to administer all drugs at the MTD. We wouldn't expect this, especially in the setting of molecularly targeted agents and immunotherapies. So we should really be thinking about redefining the criteria, used for the recommended phase 2 dose, the RP2D. Mark Rattain years ago wrote a really nice commentary about this, really kind of saying, why is it in cancer that we feel the need to define a single recommended phase 2 dose as part of a phase 1 trial, really arguing for the idea that we could come up with a range of doses, and the next step should really be dose ranging studies, where we then focus in on dose finding based on efficacy after we have identified a subset that is a reasonable subset to investigate. So, should dose response be really added more as a phase 1 question? So dose efficacy should be an integral part of drug development. It's not true that the highest safe dose is necessarily optimal. In those previous examples, you saw that Nivolumab and pembrolizumab, there was really a lack of increasing dose response relationship, and those are not the only 2 examples. Temsirolimus in kidney cancer predates this considerably, as does anastrozole in breast cancer, from an example here shown in 1996. So some proposals for change. Phase 1 trials, and I'm not saying that we should do all these are proposals for change. Phase 1 should identify a range of doses for phase 2 instead of 1 dose based on safety. Phase 2 trials should include 2 or more doses. Phase 1 and 2 should be merged using a coherent approach for optimal dosing. And phase 1, 2, and 3 should be blended for a more continuous drug development process. And this has been referred to as seamless drug development, where there's really not so much of a sort of staggered, sort of discrete set of trials that go on, but really that there's more of a drug program that is envisioned early on in the development of a drug. All right, so some statistical challenges to bring up. The Pembro and the Nivo trials, they really morphed into dose ranging studies. If you look back to those examples, they were really comparisons of patients with the same types of cancer, where they were really doing practically head-to-head comparisons of different doses of the same drugs. And they were randomized. And the results in those studies showed that there was a relatively flat dose response curve, and there was a relatively flat dose efficacy curve. So what we might ask ourselves is, how could we have designed these trials to find the best dose? What we would have needed were designs that had a variety of dose efficacy curves that allowed flat, shallow, or steep toxicity curves as well. And it'd be also interesting to think back to whether or not it was known at the time that a 3+3 design would be inappropriate to find an optimal dose. Was it really thought that getting up to, was it really thought that there would be an increasing toxicity curve and also an increasing dose response curve? Okay, so this wasn't in my initial title, but I've had the good fortune to have some interesting conversations over the past year or so about dose optimization in the post-market setting. And what this means is essentially, after drugs are approved, they are then given, and it is much better understood what their properties are and their toxicities and their effects in, quote, real world patients, and whether or not the approved doses are really the best doses for patients. And there are different ways to investigate that. And I'll talk about a couple initiatives that are really looking to think about this and implement this more. So can we optimize doses of already approved drugs? So oncologists frequently start at lower than those approved, doses lower than approved, due to experience with the toxicity profile in the real world. And there are also frequent dose modifications in certain populations. And as an aside, at ASCO, we just recently did a survey with medical oncologists, breast medical oncologists, and GI medical oncologists, and general oncologists that treat breast and GI cancer patients, and asked them about their practices with some of the most common drugs for breast and GI cancer patients. And we asked them about the frequency of their dose modifications and about starting at lower doses. Those results are being analyzed right now. We are submitting an abstract to the ASCO annual meeting, but we took this on because we know that this is a real thing that happens, and trying to get a better understanding about whether or not oncologists are doing this a lot in practice, and how we might make an impact in changing this to make care better for patients and have patients have fewer potentially side effects from therapies they receive. Okay, so Project Significant, this is a collaboration of the American Statistical Association. It's a Statistical Methods in Oncology Scientific Working Group with the FDA Oncology Center for Excellence and the Longevity Foundation, and it was organized and hosted by the American Statistical Association. So the objectives of this project they cross a number of different areas, as you can see the objectives here, but it was one of the big things was to try to bring regulators together with academicians, professional organizations, industry, and patients to have conversations about how we can have improved designs of cancer clinical trials. And in our July 2021 session, we talked specifically about this issue of dose optimization and how can we find optimal doses for drugs that have already been approved for cancer. And is it appropriate to do clinical trials in that stage, and how could we do them? So another group that's very interested in tackling this problem of dosing in cancer patients specifically for post-market drugs is the Optimal Cancer Care Alliance, OCCA. And they say that they believe it's their clinical, financial, and ethical imperative to replace current oncology overdosing with optimal dosing. The right amount of medication at the right time reduces toxic side effects, provides the best clinical outcomes and lower costs. So I say here it's led by medical oncologists. Key people involved in this are Mark Ratain at University of Chicago and Ian Tannick at Princess Margaret in Toronto, and Alan Lichter also. I think he was the previous chairman. I hope I'm not misspeaking about that. But you can see here on the right, this is from their website, and there's a nice paper from Saratella in 2020 describing this. And you can see right on here that pembrolizumab is listed, as is nivolumab, and even at the current doses that have been approved, despite all of that I showed you in terms of essentially dose ranging studies, this group believes that they are still doses that are too high for these patients. If you want to learn more about it, here's their website. So, in thinking about this, the post-market setting, we had some great discussions at Project Significant. It's a nice, although it's kind of the horse is out of the barn, it's already been approved at a dose. The nice thing about thinking about it at that time in the post-market setting is we have a lot of information already about toxicity and efficacy. But at that point, it's also the time you could think very broadly about what does optimizing a dose mean. It doesn't just mean dose. It could mean changing the schedule. It could mean changing the delivery mode. It could mean changing from a fixed to versus based on body surface area dosing. And of course, it would require input from clinical and pharmacology colleagues to determine what would be appropriate considerations for doing so. It's not going to be one size fits all, and it's going to really depend on what we already know about the drug and thinking about why are we considering a dose optimization for this particular drug, compared to all of the other ones we could be looking at. One of the things that many of you might know, where you've been involved in a situation where there's reference to de-escalation studies. This is something that seems pretty negative, and patient advocates will say, please don't call them de-escalation studies, because what that means to patients is that you're giving them less of a drug that is potentially helpful, life prolonging, or even curative. So really focusing on it being a dose optimization versus a de-escalation is important. And we've seen examples of this done in breast cancer and in HPV-related head and neck cancers, recognizing that a lot of these patients were really being over-treated for the kinds of cancers that they had. In these situations, if you are trying to consider a less intensive dose or schedule, non-inferiority trials are a logical choice, but these can be very tricky due to defining what would be an appropriate margin of non-inferiority, sample sizes would need to be large, and an optimal endpoint might not be practical in a more real world setting in this post-market setting. So there are different types of endpoints, as I just alluded to. An optimal endpoint might not really be practical. So a lot of us have been talking about different real world endpoints you might use in this setting, looking at things like adverse events. One of the things that we've been talking a lot about is dose interruptions or changes, delays or discontinuations. For many patients, if they have an adverse event, they may be more inclined to just discontinue treatment versus having a dose reduction. And we know quite well that, if we can keep patients on lower doses longer for some treatments, that having that additional drug exposure is more important than it is to have them have high doses for a shorter amount of time. So looking at how long patients stay on treatment, regardless of dose potentially, and then also drug exposure, which would really represent like a cumulative dose of drug. We can think about other endpoints like efficacy or composite co-endpoints related to composite sets of events. It is possible to do post-market randomized trials. One of the things that I think a lot of people advocate for in this setting, especially patient advocates, is to ensure that eligibility criteria will be broad. One of the really important things in the setting would be to ensure that the patients enrolled in the trial reflect the patients who receive the drug in the quote real world. That doesn't sometimes happen in the pre-market setting. And it's something that at ASCO especially, we really advocate for that, trying to keep eligibility broad. And not just keeping eligibility broad, but trying to create a recruitment strategy to get a representative sample on your study. So we know well in the April and May sessions of Project Significant, we talked about the challenges of getting representative patient populations in clinical trials. It's important to recruit patients of older ages and also race minority patients. It might be important to do pharmacokinetic studies if you're looking for dosing or schedule changes. And then lastly, primary endpoint considerations. What kind of endpoint should you have? Is it practical to do something that relies on, for example, a RECIST criteria in this setting? And if it's going to be a pragmatic trial, that might be something that is not very available. Some people have been discussing in the post-market setting whether or not there's a role for real-world data for dose optimization. It may provide evidence to support trial design. So you can utilize real-world databases. So for example, like Flatiron or CancerLink database, those are large databases of derived from EHR data on cancer patients. They can help you investigate whether or not the doses and the schedules that you're looking at may be ones that are used in practice, and maybe help you understand adverse event rates. One of the things you have to be very mindful of is that adherence in the quote real world is going to differ from protocol adherence to treatments. And this is due to a number of different factors, some of which may have to do with cost of drug. When you think about being on a clinical trial for experimental therapies, oftentimes the cost of a drug is covered, but in the real world, once it's approved, even if you have insurance, sometimes the treatments can be very expensive, and that cost benefit analysis may make people end treatment sooner. And oftentimes people will delay treatment to sort of spread out costs over time. Real world data may be lacking for traditional endpoints. So if you are using a real world database, sometimes it can be hard to identify adverse events. They are not coded in the way that we have them coded and collected in clinical trials. And recurrence, progression, or response, those are things that are more subjectively evaluated outside of a clinical trial. So many of us in clinical cancer research are used to RECIST criteria or other standardized criteria. That's not something that you're going to see in a real world data set in a patient's EHR. So the real world evidence is not really directly comparable to randomized trial evidence, due to a number of different factors, but it still can be a valuable resource for understanding what is happening to patients that are being treated off protocol. So there are a number of people working in this area of hybrid designs. I know this year at the Society for Clinical Trials meeting, there's going to be a conversation about sort of combining or harmonizing or leveraging real world data to help supplement trial data with a focus on cancer clinical trials. This can be quite complicated, and it really can be complicated by issues of potential bias. I've listed a couple examples here where people have come up with some different hybrid designs to try to really supplement essentially control groups in clinical trials by using real world data. And it can be concurrently collected data, or it could be historically collected data, the latter being more of a concern for potential bias. And then there's some thought about perhaps crossover trial designs could be appropriate in this setting. And here's just a simple example. If you're looking to see about adverse events, for example, in 2 different doses, you might randomize patients to a lower dose or an approved dose. And then you can select the number of cycles for part A based on prior knowledge of tolerance. So if you had an endpoint where you might expect to see tumor shrinkage or progression-free survival of a certain length of time, on part A, you might be able to compare that. And then you could cross over to part B where patients switch dose of treatment. This has some facets that are attractive about it. However, you would have to be careful about biases in part B endpoints if there's differential dropout in part A, and you may have and you wouldn't have any direct comparison of long-term efficacy on one or the other. The benefit of this design would be that all patients would receive the approved dose at some point in the study, which may help encourage enrollment in the study. And then, of course, the specific trial elements would be highly dependent on what we already know about the drug in question here. Okay, so I want to just talk about, I think I've given you some examples of the things that people are talking about and working on. And I think some things that are actually, really being talked about right now, really with FDA and other groups interested in making a change in dose finding and dose optimization. But there's some other things specific to dose finding and or immunotherapies that I think are particularly ripe for investigation. So some of the common themes in the examples I showed you earlier, the pembrolizumab and nivolumab, is that there was a flat dose response relationship, relatively low toxicity. The studies really became dose ranging studies. Even after those large phase 1 studies, there was still uncertainty about the optimal dose. And the way that we arrived at those final numbers was really based on haphazard dose escalation based on an MTD paradigm. So to me, this highlights the need for uptake of appropriate dose finding approaches that allow exploration of a variety of dose efficacy curves, or I guess I should just say a model that allows a variety of dose efficacy curves that would include flat, shallow, or steep toxicity curves as well. So we have a number of designs available, but what we really have, I think, is a problem of uptake of many of these designs. Something else that I mentioned earlier that I think is another area ripe for consideration is toxicity measurement. Many studies are still very focused on that binary measure of DLT. We know that patients are more complicated than that, and the toxicity experience is broader than simply a thumbs up, thumbs down in terms of what they experienced in their first cycle or beyond in treatment. Another thing to consider here is that the DLTs, well, I shouldn't say the DLTs, the adverse event profiles differ considerably with different kinds of targeted or immunotherapies compared to cytotoxic. And again, we are a bit stuck in a paradigm that was developed for cytotoxic, for chemotherapeutics, and it really hasn't proven itself to be sufficiently flexible to accommodate some of these newer types of agents. So that is something I think is worth considering. One of the things that this comes up still a lot. There's still a lot of talk about, how do we deal with the fact that when, in cancer, we're really interested in, things like overall survival, progression-free survival, and these immunotherapies are great because we have this subset of patients who are having these durable responses. But what ends up happening then is we end up, because of the way chemotherapies affect patients' cancers, and the way immune checkpoint inhibitors affect patients' cancers, we end up very frequently in scenarios like this, where we have, for the two different drugs, here we have Nivolumab and investigators' choice chemotherapy, we see almost identical or overlapping PFS curves that then separate here. And we have so long relied on a hazard ratio to reflect treatment effect, but that the hazard ratio requires the proportional hazards assumption. So we're seeing these examples, we've been seeing them for a long time now, where that does not hold. And so when you design a study based on that, and then you see results that look like this, you really have essentially violated all the assumptions that went into your your power calculation and your predefined analysis plan. So there's a need for methods that can better capture this difference that addresses the durable responses among responders, and then also addresses how to quantify overall the treatment benefit. One other thing I'll just mention briefly is one of the things that we see in with some of the immunotherapies is what's called pseudo-progression, and this is a scenario where it looks like the tumor is progressing, but really the tumor, just sort of roughly is almost blowing up. It's kind of it's having, in a sense, an increase in T cell infiltration, and it looks like the tumor is getting bigger, but really what it's doing is it's getting bigger, but it's also dying at the same time, in a sense. So there's been a challenge with trying to distinguish between true and pseudo progression, and so this leads us to wanting to have measures of efficacy that are not going to rely on specifically progressions. Now, this is relatively rare, but it's really important to understand this, because sometimes these patients that have the pseudo progressions are actually having the most benefit. And if we think it's a progression and therapy is stopped, we could be taking something away from them, when in the reality, they're having a good response to therapy. Something else that I think is really incredibly important here is that if you develop something, or even if somebody else develops something, if you find that it's not easy to use, there's a huge interest in making it accessible for others to use. I think that we have so many elegant statistical methods out there by so many smart people, but so often they're not fully developed into implementable for implementable use. So we don't have things like software, R libraries, GUI interfaces where other people can use them sort of immediately. So you want to make it easy for people to use. And I joke here, not too easy. We as statisticians want to make sure that we're involved in the trial design and the analysis. And if you make it too easy, not that we'd be out of a job, but we worry about designs that don't incorporate all the necessary assumptions. So you want to go fully forward with ensuring that the method that you develop or that you're using is able to be used by others, and that includes making documentation with good use cases for others to use. So I've just shown here, MD Anderson does a great job of this. They have a page where you can see here all sorts of different kinds of software for different kinds of trial designs, for different kinds of analyses. So I referred earlier to the FTOX design that's here, the Boyne design that's up here, and so forth. So really important to make sure that if you come up with something clever and useful, that you actually make it really useful and implementable. I know I'm just about out of time, so just really briefly, you want to abandon, a lot of people have called for abandoning traditional phase terminology. So let's stop talking about phase 1, phase 2, phase 3 trials, and think about what we're really trying to answer for each trial as drug development proceeds. There's been talk about, referring to seamless drug development. We should think about drugs being developed as a program, not these discrete phase 1, phase 2, phase 3 steps that are not integrated with each other. So, just getting back to the overall point here is, about dose finding and dose optimization. There was a very nice perspective by some people at the FDA about how they're feeling right now about dose finding, dose optimization. And I'm not going to go into, I won't read this whole thing, but the gist here is that you use small cohorts of patients receiving escalating doses and wait till they see DLTs, and then we define the MTD as the optimal dose. So they're saying here, the highlighted last sentence, we believe this practice should be re-examined for targeted drugs and biologic therapies. Basically saying we need to stop thinking that is the mold, that's the paradigm we should be using for dose finding. Okay, so I just want to revisit my 2 main points. So hopefully I've at least gotten you to consider that the commonly used dose finding designs do not find optimal doses for targeted and immunologic anti-cancer agents. And for many of the currently approved anti-cancer agents, dose optimization should be revisited. All right, so that is the end of my talk, and we have a few minutes for questions.
I have a question more, from implementation, I guess, perspective. So if one does a trial for post-marketing dose optimization, then what will it necessarily lead to some updated regulatory guidance, or how that then will be implemented into the actual clinical practice?
Yeah, it's a good question. I think it would depend on who was doing the dose finding or the dose optimization. So for example, if there is a group that decides to do a pragmatic study, for example, this would be something, if you're familiar with the PCORI, they might be interested in finding something that they're very interested in patient-centered, patient-focused research. It could be such that findings are reported that lead to a change in clinical practice, but not necessarily a change in the label. Or it could be such that if the drug company were so moved, they could apply for a change in the label if they wanted to, but that's not something that would necessarily happen. But I think the important thing is doing the study and then disseminating the research to ensure that the guidelines be updated if there are guidelines, and that care providers understand the pros and cons. And it might be a question of precision medicine, right? It could be such that older adults over age 75 should be started at 1 dose, and patients under age 75 generally started at a lower dose.
Thank you. So we also have a question from Meredith, and you should be able to unmute. And
yeah, I'm good. I'm unmuted. Wonderful talk. Excuse me. Wonderful talk. Thank you so much. I'm very curious in thinking about this post-marketing setting, specifically for dose optimization. Isn't something I've really thought about before. And I was wondering, in the discussions, especially with Project Significant, have they been really thinking about this issue of long-term or late emerging side effects, specifically of IO, as it's starting to be introduced into the early disease setting where you're curative, and the idea that you could have these very late and persistent endocrine side effects coming from IO? Is that a topic you've discussed?
It hasn't been a topic, I would say, that has been discussed specifically. You might be able to tell from that group. It's been quite broad in terms of representation across the world, but at the same time, it's been a lot of it has been methodologists and from academia, obviously me from ASCO, and regulators, and we haven't been very specific really at all about individual drugs or even areas, but I think the whole reason it has come up is exactly for that reason. You know, now that these drugs are in the post-market setting, we're recognizing that there are a lot of maybe not so much subtleties, but issues in different patient populations for which we know relatively little. And the fact that, you know, the studies are designed with relatively short follow-up. I think it's a great point. And I think you're exactly right to be thinking about patient populations who, you know, all the ones that I showed you for Nivo and Pembro, those are patients who had metastatic disease. And if now we're talking about bringing these drugs into early stage patients who would be expected to have, you know, very good long-term prognosis, that those would be, you know, real concerns about how we're going to follow them long-term and quantify their potential for adverse events.
Great, thank you so much.
So we have another question in Q&A from Wendy London. And she asks, are you aware of any discussions at the FDA that are specific to pediatric oncology dose finding?
Yeah, that's a great question, Wendy. And I am not aware of any specific discussions about pediatric trials, but certainly, you know, great thought, great idea. And that's, you know, certainly a patient population that needs to be considered.
The question is, will the slides be shared? And Pearl Sampson says, thank you for the great talk.
Yeah, I'll be happy to email them to, I guess Erica is whose email I have, and she can share them if that's appropriate. Or I don't think I put my email anywhere, but I'm happy for my email to be shared.
Also, this talk was recorded, so it should be appearing on the department YouTube channel. So if you would like to rewatch it.
Thank you, Liz, and congratulations on your new leadership with
Thanks.
ASCO.
Sorry I can't see you in person, everyone,
No.
especially including Meredith and Wendy, and maybe others that I know and I haven't seen their name on the list. 2023, we'll get together. Maybe 2022, we'll get together at the ASCO annual meeting.
June. Yeah, it was really excellent. Really covered a broad variety of, a uniform but broad breadth of topic, which was really phenomenal.
I mean, I know we're just at time, but I will say leaving the academic nest and finding myself at ASCO has just been really interesting to think about and be exposed to, be a different stakeholder in a sense in a lot of these discussions. So it's been a lot of fun having that perspective.
Right. Thank you very much for enlightening us on important topics, and it was a great pleasure to have you. Thank you everyone for attending.
Thank you.